I've known her for 9 years now, and it was a very nice time in her group. It was a very happy group to be in. And I could say that I learned from Inês, not only academically, but just in general, many things beyond academically. So it was a great experience in her group. So I'm very happy to have Inês here today to tell us about her research in the genomic architecture of glycemic traits across multiple ancestries.
Great, thanks very much, Jen, for the really nice introduction. Hopefully you can all hear me okay? Great. So, yes, over the next, hopefully 50 minutes or so, I'll be telling you a little bit about the work that we've been doing, understanding the genomic architecture of glycemic traits. So just to kind of get everybody in the same frame of mind, what do I mean by that? And what are glycemic traits used for? So most glycemic traits are used to either diagnose or monitor diabetes, i.e. how well do people respond to anti-diabetes drugs, for example. And there also can be very helpful traits to study to better understand pathways that relate to healthy human metabolic physiology, as well as for those people that do have diabetes to understand pathophysiology. So for those of you that are not familiar with type 2 diabetes, most of my talk pertains to glycemic traits mostly used in the context of type 2 diabetes. So type 2 diabetes really occurs due to two different things. On the one hand, we have insulin resistance, which means that the insulin target organs, mostly muscle, liver, and fat, are less sensitive to the effects of insulin in terms of uptaking glucose from the bloodstream and returning glucose to normal levels. And that means that we need higher concentrations of insulin for it to have the same effect in terms of glucose lowering. That in itself puts some pressure on the beta cells in our pancreas to increase insulin production. And if there's a beta cell deficiency so that it can't continuously increase insulin production, that's when type 2 diabetes occurs. So you have an inappropriate response from the beta cells in terms of your insulin resistance. Now, type 2 diabetes has historically mostly been diagnosed on the basis of the two traits. Mostly fasting glucose. So this is when doctors ask you to have your meal and then fast overnight, don't eat for 8 hours, and then in the morning they call you in, take a blood sample and measure your blood glucose. So that's your fasting glucose. And you can see on this plot here, hopefully this pointer works. Yeah, in a healthy individual, their baseline fasting glucose is lower than somebody that has type 2 diabetes. And this measurement has been used to diagnose individuals with type 2 diabetes. Another measure that is routinely used to diagnose diabetes is 2-hour glucose after a glucose challenge. So this is, again, you come in fasting overnight, they give you a glucose syrupy drink to drink, maybe something like Glycated, and that will cause a spike in your blood sugar. And in a healthy individual, you see the spike, then at some point insulin gets secreted and your blood sugar levels will start to return to normal. Actually, at 2 hours, they're slightly below your baseline. And what you will see is the type 2 person, that kind of response back to baseline is a lot slower. And these have been traditionally the two measures used to diagnose diabetes. The third measure that is used is something called glycated hemoglobin, and that's a little bit more complicated. So glycated hemoglobin is a biomarker that is, as the name indicates, hemoglobin, which is the protein in the blood that it carries oxygen, and it results from a non-enzymatic reaction that glycates the hemoglobin, and it actually measures proportion of your hemoglobin that is glycated. This reaction is believed to be irreversible, and therefore it reflects average blood glucose concentration throughout the lifespan of the red blood cell, which is about 120 days. So when somebody measures glycated hemoglobin, they're actually measuring average glycation in all ages of red blood cells that we have in the blood. And there are a certain number of assumptions that are made. A, the reaction is irreversible. B, most people have the same lifespan of their red blood cells, etc. But that is what is used to determine glycated hemoglobin. Now, because you don't have to be fasted, and because rather than it giving a snapshot of what is your glucose at that point, which is what fasting glucose and 2-hour glucose measure, it's actually become the most prevalent method to diagnose diabetes because it's more convenient and actually gives you a window into a 3-month period. How well are you controlling your glucose? And actually, for a very long time, glycated hemoglobin was actually used by doctors to try and understand how well patients were responding to their diabetes treatment, or indeed whether they were taking the anti-diabetes treatment, because they gave them the possibility of detecting the patients that lied about the fact that they actually hadn't been taking their medication. Their fasting glucose might have been fine that morning, but their glycated hemoglobin told the doctor something quite different. Anyway, so these are the traits, some of the 3 traits that we work on, and the other trait that is worth talking about is fasting insulin. So as I said, insulin is basically the hormone that is secreted from the beta cells in the pancreas. At baseline, again, after overnight fast, it will be somewhere there. When you have your glucose syrupy thing, there'll be a spike in insulin secretion, and then there's a second phase insulin secretion. So we measure fasting insulin. And this measure is an imperfect measure, but is somewhat of an assessment of insulin resistance, because at baseline, if you have very high fasting insulin levels, it usually means that you need more insulin for it to maintain your just baseline glucose levels. Now, type 2 diabetes itself is quite variable among populations across the world, and its underlying risk factors also vary in their prevalence across the world. Having said that, as for most other complex traits, we've been rather limited in our ability to study genetic studies of diabetes and related traits in very diverse populations. So what I'm going to talk to you about mostly for the rest of the talk is a study that we did over an embarrassingly long period of time, which I won't tell you, but was finally published last year, where we basically tried to increase diverse representation in studies of glycemic traits. And then we had specific aims to explore how having a more diverse population in our genetic studies would impact our ability for locus discovery, tell us something about how transferable the loci were across populations, and also about how this might impact our ability to find map our association signals. This was done within the context of a large international consortium called MAGIC, which was our first big meeting, was how do we call ourselves? The most important decision of the consortium was name ourselves something catchy and find a nice colleague that was artistically talented, definitely not me. And we've been going for a long time. I think our first paper together was 2009. This is our website. We put all our data up publicly, summary stats available for anybody to use, and they're also all in the GWAS catalog. So for this particular study, we set out to try and harness as many different cohorts as possible that had 4 traits: fasting glucose, HbA1c, fasting insulin, and 2-hour glucose. The number of cohorts that contributed varied quite a bit, from 41, which is the smaller sample size we had, was for 2-hour glucose, which was just shy of 86,000, with fasting glucose and HbA1c having the majority, the larger sample size, fasting glucose almost 300,000 samples. Despite our best efforts to increase population diversity, we still only managed to get about 30% of our samples overall being from individuals of non-European ancestry, mostly represented by East Asian, Hispanic, African American, and South Asian. And for HbA1c, we also had a cohort from Uganda, which had about 7,000 participants. As most large consortia that don't have access to underlying individual level genotype data, there was a lot of local analysis by cohort analysts that did their own individual analysis, quality control, and imputation. This was all imputed to 1,000 Genomes Reference Panel at the time. This predated TopMed and other things. They conducted their own GWAS, and then there was a central group that did some more quality control. And we conducted meta-analysis first by self-identified ethnicity and verified by genetically clustered individuals. So we did European and each of the other ancestries individual meta-analysis. and did kind of approximate conditional analysis using GCTA, and then we used a cross-ancestry meta-analysis program called MANTRA to find variants that harness the entire data set. Now, this posed a lot of challenges, and one of them was, how do we count our beams, i.e. our loci? And the reason is because you get lots of different top signals. When you just have one trait and one ancestry, you take the SNP that is the most significantly associated, and you tend to cluster your SNPs by some combination of LD and distance and say everything within this interval is my locus. But here we had a trans-ancestry lead variant, which might be different from the South Asian lead variant, and then I might have another lead variant. This might be 2 for fasting glucose, and I might have a 2-hour glucose just here. So we spent hours deciding how are we going to call these and whether we would group them or separate them. And there's like a whole textbook in our papers to explain what we call the locus. Don't worry too much about it, because that's just to count, and we didn't want to overcount. So I was quite adamant that I didn't want to count fasting glucose separate from 2-hour glucose, because these traits are highly correlated, and it's quite likely, even if the lead variant is slightly different, if they're mapping quite close together, they're probably tagging the same underlying causal variant. So anyway, we basically ended up with some definition that started one end of the chromosome and merged together things. As long as they were within 500 kb of each other, we kept grouping them into a locus. That also helped when it came to fine mapping. We didn't artificially break 2 things and therefore not allow us to find that they actually shared the same causal variant. So this is a long-winded thing just to say our locus definition was very complicated, and if I was to do this all again, I think I'm just going to drop the whole locus idea, because I don't know what locus means anymore. And if any of you out there have better ideas, I'd really welcome your thoughts on that. So we found lots of loci, as you might expect. We found 242 loci, 99 of these were novel, and here at the time we defined novel as in within this locus, there was no established signal for either any of the glycemic traits that we were looking at, or indeed type 2 diabetes as case control signal. So these loci mostly were identified across several ancestries, and then we had 7 that we're calling single ancestry. I'll return to that, but just to be clear, single ancestry doesn't necessarily mean that the SNP is only seen in one ancestry. The SNP might be polymorphic and seen across multiple, but the evidence for association is driven by a single ancestry. So what did we find? As we expected, these traits are quite correlated. So the loci, there's quite a lot of overlap between the loci. There are some signals that appear to be unique, or loci that are unique to some traits. We do have to be a bit cautious how we interpret this, because if you remember back to one of my early slides, sample sizes between these traits vary quite a bit. And so, of course, it might not be genome-wide significant for 2-hour glucose just because I don't have power for it. So I don't read too much into this. And in fact, this was just, some people like counting things, and they always want you to have these counts in your papers. So we had the counts there just to, tick that box for the reviewer that wants to count. But what we were really interested was trying to kind of also assess what did the fact that we had diverse populations, how did that contribute to our locus discovery? Now, as you expect, when you do a multi-ancestry meta-analysis, you find more things, but obviously you're also harnessing all of your power from all the samples. So what I've tried to show here are, for each trait, the number of loci that came out as genome-wide significant in the trans-ancestry meta-analysis. And as I told you, European was by and large the biggest slice of the pie. And so of these 100, 18 hadn't been genome-wide significant in the European data alone. But again, the European data alone, although it's about 70% of the sample, is not the entire sample. So what we did was we tried to emulate the sample size in just Europeans to be the same as the trans-ancestry by rescaling the standard errors of our associations in the European data. And we found that if we did that, we'd actually, had we had the same sample size that we did in the multi-ancestry analysis, just comprised of European participants, we would have found 30, or actually 29, because there's one double counted, 29 fewer loci than including the diverse populations. And of those, 24 were novel. So here's an example of what I call the single ancestry locus. It's a fasting glucose locus. That's the index variant. It was driven by data from the African American participants. And the minor allele frequency is actually very similar in both European and African ancestry individuals. And the reason I flag this is for the most part, where people talk about ancestry-specific variants, the reason why we find it in one ancestry and not another tends to be because the allele frequencies differ a lot between the ancestries. And that wasn't the case in this example. The effect size is actually very different. So obviously there's no evidence for association in Europeans, and there's a reasonably strong effect in the African Americans. And the nearest gene actually turned out to be fairly plausible. So it actually codes for a transcription factor, which is expressed in the beta cells in the pancreas. It co-localizes to insulin in the beta cells. And actually, when you challenge cells with a high glucose and you have overexpression of this gene, you actually inhibit insulin secretion. So overexpression of this gene seems to be involved in glucose-stimulated insulin secretion. So it seems quite plausible. Now, why would we find a variant that is present in both ancestries and is associated in one and not another? I mean, ultimately, we think biology is the same, irrespective of how unmixed we are and what population we come from. But ultimately, I guess we put it down to one of three things. Either there are some gene-gene interactions, gene-environment interactions, or the variant that we find in African Americans is tagging a lot better some other true underlying causal variant that isn't well tagged in the Europeans. We tried to look for rarer variants in the African American ancestry that might explain the association, but we're limited by variants that were in the 1,000 genomes, so we couldn't find anything, but that's still a possibility. So the next question we wanted to try and answer was, how transferable are our loci across the various ancestries? And we looked at this in two different ways. One was we wanted to understand what was the correlation of the allele frequency across the different populations. And very much in agreement with population history and evolution, what you can see is that there's a high degree of allele frequency correlation between Europeans and Hispanics, Europeans and South Asians, African Americans with the Africans, and so forth. So this all kind of made sense. But then the more interesting question perhaps was this question about how much evidence do we have that, given that the locus is shared, there's evidence for allelic heterogeneity, or heterogeneity of the effect allele, I should say. So I don't expect you to see the numbers that are here, but actually, again, I'm comparing between the different ancestries and the different traits, whether collectively the loci have evidence for effect allele heterogeneity. And actually, most of these did, but when we did sensitivity analysis and we removed by one the variants that did themselves have evidence of heterogeneity, what we find is actually overall 80% of the loci had no evidence of allelic heterogeneity, effect size heterogeneity, and there's about 20, but actually the flip of that is that 20% did have evidence that there was some heterogeneity of effect size across the different ancestries. And actually the traits that has most evidence for evidence of heterogeneity in effects allele is HbA1c, and I think I know why. I'll come back to that one later because I'll have to explain something else about HbA1c. So hold on to that thought or not. So one example of a variant that did have evidence of between ancestry heterogeneity is this one here. Again, you can see the effect allele frequencies down here for each of the ancestries. And again, the African American has a very strong, the effect sizes are much larger and there's no evidence for association at the others. And for those of you that are skeptical about this, which I was, this isn't driven by a single cohort. We drill down to the data very carefully. There's several cohorts of the African American ancestry that contribute to the signal. It looks very compelling. SNPs are well imputed, all these various things that you would look at to make sure that that's not a spurious association. So in terms of variance explained for each of these traits, we still actually only explain a relatively modest proportion if we just take the genome-wide significant variance for each ancestry and each trait. And as you might expect, given data that has been published by many others before, if you actually take the signals that come out of the multi-ancestry analysis and you add in the ancestry-specific ones, by and large, you increase trait variance explained. In other words, even though many of these SNPs are not genome-wide significant in those ancestries, they're still contributing to the genetic architecture of that trait on that ancestry, and probably the reason why they're not genome-wide significant is because we're really underpowered. No surprises there. So polygenic risk scores, we weren't really going to do that, but the reviewers forced us to, so we did. So we kind of thought, well, this has been done by many others before, but fine. So as you'd expect, and in agreement with what many others have shown for many other traits, if you have a polygenic risk score, in this case, we did one with PRSCS, which is one that allows us to just have the polygenic risk score based on summary stats, and also that had reference panels for several of the ancestries that we were working with. You can see that obviously if you build it based on Europeans, the trait variance explained does better than if I just take my genome-wide significant variance. Okay, so no surprises. But also that it does much worse in the other ancestries. Again, no surprises, it's been published many times before. But we were asked to do it, so we did. So, how does diversity then help with fine mapping, or does it? So to do this, we performed fine mapping using FineMap, and one of the advantages at the time when we did this was that FineMap doesn't actually assume that you have a single causal variant at the locus. And so the first step, it actually estimates how many causal variants it thinks it has at that particular locus, and it's still estimated that about 61% of our loci were due to single causal variants. And then when we did the fine map, we have 21 loci where the credible set, the 99 credible set, is reduced to a single variant, so that's pretty good. And there's a reasonable number of locus trait associations where the credible sets had 50 or fewer SNPs, which we drilled down more carefully to try and identify co-localized EQTLs, coding variants, all those various things, which I won't go into details here. And one of the things I will say is, why am I calling this locus trait? Is because if I remind you, our loci actually have multiple traits. So for each locus, you have to find map it for each trait. This was before we started working with Jen and trying to do it across multiple traits, which will be a next chapter, which I don't have the results for yet. So one of the loci where the credible set was comprised of a single variant is this one here. FineMap identified this SNP as having a high posterior probability of association of 0.993. This looks really compelling. This SNP actually maps, co-localizes as an EQTL for the expression of this gene here called insulin receptor. The name is already quite telling. It basically codes for the receptor of the insulin gene and it basically starts the cascade in insulin target organs that is responsible for translocating the glucose 4 transporter to the cell membrane and taking up glucose. And very nicely, the direction of effect makes perfect sense. So the allele that is associated with higher fasting insulin is associated with lower levels of expression of the receptor. So there's some level of insulin resistance in these individuals, hence the higher insulin. So overall, if we then compare fine mapping results with done in single ancestry on the x-axis versus across ancestries on the y-axis, you have a number of loci that are kind of highlighted in gray that don't do any better. Some do worse when you include the multi-ancestry data, but the vast majority improve. But of course, again, the question is, does it improve because we're leveraging all the samples for fine mapping, or does it actually improve because of differences between the populations? So to try and answer this question, we did the fine map in the trans-ancestry analysis and in the single ancestry using the exact same SNPs that went into the fine map, so that wasn't introducing any variability in the results. And again, we kind of tried to emulate the sample sizes by rescaling standard errors. And by doing that, we figured out that about 50% of the loci have improved resolution just because of the increased sample size in the meta-analysis across multiple ancestries. But actually, for another about 50% of those that get better, that's because we're leveraging differences in LD that exist between the different populations. So just to kind of summarize this part of the talk, what have I told you so far? Based on the data that we had for these different glycemic traits, we think increasing the diversity helped us discover more loci. It allowed us to explore the transferability of the loci and indeed demonstrate that about 80% of loci transfer with no evidence of effect allele heterogeneity. The trans-ancestry discovery, including those SNPs, does increase trait variance explained even if they're not genome-wide significant across all populations, and there is evidence that it does improve fine mapping resolution. You probably all think, well, that's not really surprising, but at least we could show it. So now I'm going to tell you a little bit of a different side story or vignette and go back to HbA1c. So HbA1c is quite a complicated biomarker, which I've been studying it for a while, but the more I study it, the more I think it's complicated. So anyway, HbA1c is basically glycation that occurs in the red blood cells. So for those of you that aren't familiar with red blood cell kind of development, which I wasn't, they start off, let's say here, as a reticulocyte, a young red blood cell. And the glycation occurs throughout their life as they mature. And as I said, this is called glycation rather than glycosylation to accentuate this is non-enzymatic. It's non-enzymatic, so the amount of glycation should be directly proportional to the glucose circulating in the blood. And it is also dependent on this idea that everyone has more or less the same lifespan of their red blood cells. Now, having said that, there are situations where it is already well known that you shouldn't use HbA1c to diagnose diabetes. And in previous genome-wide association studies that we've done quite some time ago, we are actually able to demonstrate this looking at the genetics of this particular trait. So when we find genetic variants associated with HbA1c, really what we're hoping to get, because HbA1c is a biomarker for glycemia, is we're hoping to identify variants that somehow affect biological pathways relevant to glycemia and that they ultimately affect glycemia, and that's what we're measuring. But of course, there's an alternative path, which is genetic variants can affect things that have nothing to do with glycemia, but have a lot to do with red blood cell biology, structure, lifespan. And for example, it is well known that individuals that have, for example, hemolytic anemias, which is basically where the red blood cells live shorter amounts of time, you shouldn't use this assay. And the reason for that is because obviously if your red blood cells don't live 120 days, you have a higher proportion of younger cells that haven't been in the blood long enough to be glycated. And so you get an artificially low measurement of HbA1c compared to somebody else that may have the same glucose, but their red blood cells live longer. So that's fine. But what wasn't known, and something that we published a few years ago, was when we did our first multi-ancestry analysis of HbA1c, we were quite surprised because in African Americans, we found one particular variant that is on the X chromosome, so hence I have the females and the males, that has actually, for those of you that aren't clinicians, this is a massive effect size for a single variant. And this individual variant alone, we were fortunate to have individuals that had this measure, HbA1c, and also had other measures of glycemia in the same individuals. And we were able to demonstrate that even though the individuals that carried this particular variant had very different levels of HbA1c, their glucose was not different, i.e., we're seeing the effect of a genetic variant that lowers HbA1c by quite a large amount, and this lowering is independent of glycemia. And of course, the consequence of that is that we will misdiagnose individuals who truly should meet criteria for type 2 diabetes, but their measurement is artificially low. So this is obviously a problem because you're missing diagnosis, but also is a secondary problem because this variant is very prevalent in African Americans. It's got a 21% minor allele frequency. It's virtually absent in Europeans. And so you are increasing health disparities because not only are you misdiagnosing, but you're affecting one ancestry and not the others. And the reason why this happens is because it turns out this variant that maps to this gene here, called G6PD, this stands for glucose 6-phosphate dehydrogenase deficiency. Long thing, but basically what this is, it's actually a variant that causes a form of hemolytic anemia, but it's silent. So most people that have this variant do not know they have a hemolytic anemia. So their doctors wouldn't know that HbA1c is a not appropriate assay to use to diagnose diabetes in them. And the other reason why this is very common in individuals of African ancestry is because this variant actually is known to protect against severe malaria. So it's been selected for in the balancing selection. And we've actually recently gone on to look at this variant in the UK Biobank, and actually this is present in Biobank in 31% of African ancestry individuals are carriers for this. And again, we see a very strong effect. This is their effect size in terms of HbA1c, and this is comparing, Biobank doesn't have sophisticated measures like fasting glucose, but it has random glucose, and it also has time since last meal. So we can actually devise a proxy for a fasting glucose by saying, I'm only going to look at individuals that have a fasting time greater than 5 hours or so. If you really are very stringent, you lose the majority of the sample. But if you go for a compromise like 5 or 6 hours, you have enough samples to basically demonstrate the glucose by genotype is the same, but the HbA1c varies a lot. So again, this will be problematic because in the UK, HbA1c is nowadays the way that doctors diagnose diabetes. They don't use fasting glucose, they don't use 2-hour glucose. So actually part of the work we're doing right now is to kind of develop more precision diagnoses that takes into account some of these things. So I kind of jumped the gun. Why is this important? Obviously, if the HbA1c measurement is independent of or not accurate reflection of glycemia, you can either underdiagnose and undertreat, or on the other hand, there are examples where it increases the estimate. So you'll be doing the opposite. You're overdiagnosing and overtreating. So when we did our study, we then knew about these variants and thought that we would try and classify how the HbA1c associated variants might be affecting HbA1c, whether it's through glycemia or through other things. So we leveraged association data from 3 glycemic traits that were included in our study: fasting glucose, fasting insulin, 2-hour glucose, and a bunch of red blood cells that had been published, as well as iron traits, because iron deficiency anemia also affects HbA1c. It actually does it the other way. People that are iron deficient have a higher HbA1c than their glucose would suggest. So Ji Chen, a postdoc in my lab, performed some fuzzy clustering approach that took into account the association results. So basically for each SNP, we looked at the association for all these traits and used this clustering approach that allowed us to group SNPs into groups, but also we did kind of both a hard clustering and a soft clustering that allowed SNPs to belong to more than one cluster. And this, for me, was important because I think biology, there's plenty of evidence of pleiotropy, and there's no reason why a SNP might just be affecting glycemia. There may be SNPs that affect both, and we certainly found some of those. And then to confirm the label for that cluster, we took advantage of some of our samples having both HbA1c and fasting glucose measures or hemoglobin, and we could do HbA1c conditional on fasting glucose and see how that affected the effect size to kind of further explore whether the mechanism of association with HbA1c was mediated through fasting glucose or hemoglobin. And we could also look at type 2 diabetes association results. Again, a SNP that affects glycemia should associate with type 2 diabetes. There's a health warning here because, as I've been saying, the problem with type 2 diabetes associations is that historically, most studies don't tell you how diabetes has been diagnosed. So, of course, if diabetes has been diagnosed with HbA1c, you're kind of in a circular problem, and I'll show you results that speak to that. So when G did this, we actually found, much to our surprise, that the majority of our association signals with HbA1c, only about 24%, appear to actually affect glycemia itself. and the vast majority of them seem to affect mostly red blood cell, reticulocyte, and iron. I should say, and I should have said this at the beginning, one of the things that is really important is in these studies, we are not studying anyone that has a diabetes diagnosis. And this is important because obviously once somebody has a diagnosis of diabetes, they will change either their behavior or they will be on treatment that affects HbA1c. So that's no longer an appropriate readout of where they would be naturally in their biology. So it could be that the reason why we see many variants affecting HbA1c through non-glycemic is because we're actually studying HbA1c in the healthy range, not in the disease state. And actually, I've done some work since then, and if you look at what factors explain the variance of HbA1c in a population, and you look at a healthy population, and you include the top end of the distribution of those that have diabetes but don't know it, you actually see that the variance explained for those that qualify as having diabetes, so they're above a certain threshold, about 70% of the phenotypic variance is due to glucose. And in the ones that don't, quite a lot of the variance is due to things that have nothing to do with glucose. Glucose explains probably less than 10% of the variance there. So there's something that happens as you get to diabetes that affects what that measurement is actually measuring, I suppose. So we then constructed stratified genetic scores of these HbA1c variants into these different groups, depending on their labels. And this is actually based on a study that we did prior to this GWAS, where we had fewer variants. We had 50 variants. If you take overall all the variants without stratifying, this was actually looking at a score of all of these variants and risk of coronary artery disease that's very strongly associated. If you stratify them into glycemic and the kind of red blood cell related, actually both of them are associated, and in MR analysis, both of them are causally associated with increased cardiovascular risk. And this is quite interesting, because most people thought that only the glycemic ones would increase your risk of cardiovascular disease, but actually it's very consistent with what people have been studying red blood cell traits that actually have demonstrated that things that affect red blood cell traits themselves are risk factors for coronary artery disease. So actually in a very kind of more circuitous way, we found variants that affect HbA1c through this mechanism, and they too are associated with cardiovascular disease. And that may be a reason why in epidemiological studies, quite often people have found that HbA1c is a better predictor of coronary artery disease comorbidity than, for example, fasting glucose. And it may be because it's actually measuring both the effects of glycemia and something else that the glycation is doing, or the red blood cells. So we then looked at genetic scores and how they're associated with type 2 diabetes instead of coronary artery disease. And obviously, if you build a score of all of our HbA1c variants, it's very strongly associated with type 2 diabetes. And then we kind of break it down. Well, if you just have the hard clustering glycemic variants, that is responsible for most of the association. But you actually see there's a still strong association signal coming from what we're calling the non-glycemic and the hard clustering. But actually, if you break it down, it seems that most of the signal is because they are red blood cell and they also fall in a glycemic soft cluster. So they actually seem to be doing both things. So this classification is useful because what I'm going to tell you next has to do with going back to our 4 glycemic traits and these associations, kind of trying to explore what do they tell us about similarities and differences between the traits in terms of epigenomic landscape. So as for most complex traits, our variants mostly map to the non-coding region. And so we looked at a number of different features in the genome. I would say kind of the more the features that are the same, so 5' UTRs, coding, etc., that don't change by cell type, and then some features that are more specific to different cell types, such as, for example, stretch enhancers. So these are non-coding regions of the genome. They have regulatory function, and they are responsible for regulation of gene expression in a tissue-specific manner. And what I'm showing here is that fasting glucose, HbA1c, trial glucose, and fasting insulin, if we take the loci that are associated with these traits, there's very strong enrichment for signals that are enriched in stretch enhancers in islets, as you might expect, because islets are responsible for a lot of the beta cell function and insulin secretion. Very interestingly, and it kind of maps back to what I was telling you about the HbA1c trait, is for HbA1c, we also see enrichment in a cell type that is derived from blood. And actually, when we now partition HbA1c into these classified classes, you see that in the stretch enhancers, all of the signal in the islets is driven by what we're calling our glycemic variants, and the signal of stretch enhancers in this blood cell type is driven by what we're calling glycemic variants that affect HbA1c through red blood cell biology. And then interestingly to us, the fasting insulin actually was enriched also in stretch enhancers in skeletal muscle and adipose, and these are insulin target organs, so it all kind of made sense. We're now finding variants that are in regulating genes that are expressed in insulin target organs. So the next thing we wanted to explore is there also evidence that we see enrichment for loci that might be expressed differentially across different tissues? So this was work that was done by Gail Mahen, who's a postdoc in my team and now is an independent PI in Prest. And we did this using the DEPICT program that actually uses expression data, so it's not biased by some of the studies that were motivated by GWAS. And very much in agreement with what we saw with the stretch enhancers, if you took the fasting glucose loci, they're very heavily enriched in genes that are expressed in the pancreas. The fasting insulin, actually the genes that came up most strongly were enriched for genes expressed in adipose and fat tissue. I don't know what the difference between adipose and fat is, but anyway. And adrenal glands, which was quite interesting because there's some link between the adrenal gland and cortisol expression and insulin resistance. And HbA1c, very much in agreement with what we'd seen in some of our other analysis. We see the immune system and blood, and also liver and pancreas, kind of corroborating that this is quite a complex trait that kind of brings together the blood and the metabolism. So then, based on this, we did some pathway and gene set analyses. And again, this was actually done by Ali Wheeler, who's a staff scientist with me, and then moved on to the MRC EPI unit. And for fasting glucose, our variants are very strongly enriched in kind of forming glucose homeostasis, and obviously, and very interestingly, groups of genes that are associated with maturity onset diabetes of the young, which is an autosomal dominant form of diabetes that is non-autoimmune mediated. The fasting insulin was really interesting because actually it highlighted loads of pathways related to growth and cancer, and as well as metabolism, as reproduction, which there's very strong links between insulin resistance and in women in particular, polycystic ovary syndrome, also associations with cancer. And so we've actually explored this more detail with some colleagues in the cancer field. I'm jumping the gun, so I'm coming back to that later. Hold on to the cancer thought. So the HbA1c variants, again, basically tell us the same what I've been showing you before. There's loads of complicated pathways related to hematopoiesis, but also glucose metabolism. And then if I show you these results in a very non-scientific but visual way, I hope you can see that basically what comes out in very strong in each of the traits is actually quite different. So fasting insulin, all to do with insulin hormone, metabolism, glucose. Fasting insulin, a lot to do with development, cancer. And HbA1c, actually the spleen, erythrocyte come out as very, very strong. So going back to this link between insulin and cancer, it's been known for a long time that there's a link between insulin resistance and even diabetes and some forms of cancer. But actually, it's been quite difficult to know if this is mediated through glucose toxicity type of effects or whether it's mediated through insulin. And so very recently, with some colleagues in the cancer field, we have taken all these genetic scores for all these different traits and conducting MR analysis have demonstrated that actually it's a higher fasting insulin levels are associated with increased risk of colorectal cancer And actually, we don't see very strong evidence for the other glycemic traits increasing risk of cancer. So they're teasing apart the effects of glucose from the effects of insulin. And they are actually now expanding this to other cancers as well. So just to finish up my talk then, I hope I was able to convince you that using this multi-ancestry analysis, we found lots of different loci. We were able to demonstrate that 80% of them didn't have any evidence of between ancestry heterogeneity. That actually, had we had the same sample size but only European ancestry individuals, we would have found fewer things. And also the fine mapping resolution was improved independently of sample size. And there was a median reduction of SNPs in the credible set, 37.5%. And you probably will go away thinking HbA1c is a bit of a headache, but hopefully if the only thing you take from this talk is HbA1c is quite a complicated measurement, I've done my job. So yeah, it seems to be impacted by lots of different things and it's quite an interesting trait. So I think one of the things this also allowed us to do was actually not only explore what the effect of the different populations had done, but also explore more about similarities and difference from all of these glycemic traits that people tend to think, oh, well, it's all a glycemic trait, but actually they tell us a bit different stories about underlying biology. And actually they are routinely used in people that start from studying diabetes as yes, no, case control. If you can harness then information from these traits, people are now using this to try and subcluster and try and identify, because type 2 is quite a crude thing. You diagnose people on the basis of these measures that I've told you, but there's lots of different ways that people could get there, and hopefully some of these traits can help tell us a little bit more about the detailed biology underlying that. So this was a massive effort by loads and loads of people, so I don't have, it would be like 10 pages of acknowledgements if I listed all the authors. There was like 409 on that paper. So this was the key kind of A-team. So they did a lot of the heavy lifting. I think I flagged most of the key people as I went along, thanks to all the cohort PIs, analysts, and all the participants. And a plug that we're starting a new effort. And if you do have relevant data, please reach out. We're always interested in expanding our collaboration. Thanks very much for your attention. Happy to take questions.
Hi, it's a great journey through magic. You talked about the fine mapping using your trans-ancestry meta-analyses results. How did you actually do that, given that you need an LD reference panel and there's no such thing as a trans-ancestry reference panel?
Yeah, so we had to use each of the reference panels for each of the ancestries, and we weighted them by the proportion that they contributed to the analysis. So it was, you're right, it's very difficult to do that
and that can induce false LD.
Yeah.
Did you run any checks just using, for instance, just a European reference just to see if you got the same or roughly the same results?
Yeah, we did fine map in each individual ancestry and then across ancestries, and where the results didn't kind of tally up, we kind of tried to look at why that was and what was the reason behind it. Yep.
Thank you, that was brilliant. Thank you very much. There's a question back on the ethnicity or identifying ethnicity specific genetic effects. So there's some gene environment literature around the use of marginal pre-screens, where you would say pre-screen genetic variants for correlation with environment, blinded phenotype. And it can be shown that that is a kind of an independent screen for subsequent gene environment interaction testing. And so the analogy here would be pre-screening genetic variants for heterogeneity across ethnicity. So it was interesting that you showed a kind of counterexample of that and an example of that. So I was just curious if it's something you thought about or you tried, or you think it would be a bad idea given.
Yeah, no, we haven't thought about it and haven't tried that. It's an interesting thought. Yeah.
Okay, we've got a question. Sorry, we've got a question on Zoom from Vincent. Vincent's got his hand raised. Vincent, would you like to unmute and say your question?
Yeah, can you guys hear me?
Yeah.
Thank you very much. So following on the data you showed suggesting that quite a bit of the HbA1c effect are not directly affecting glycemia, it looks like HbA1c is not a perfect biomarker for diabetes type diagnosis. So how big of an effect is that in the general population? And assuming it's big enough, is there any scope to actually implement those corrections in practice to make it a better biomarker? And how do you think it's possible to achieve that?
Yeah, I think that's a really good question. So I think I should have said that. I think for the most part, it is a good biomarker for most individuals, but there are going to be examples where there are some genetic variants that have big effects. So I think for the ones that we know already, it's very easy. So in fact, you can adjust for that genetic variant and say you lower the threshold for those individuals. And so some of the work that we're doing right now is thinking about more precisely defining diagnostic thresholds. So instead of, and in fact, I didn't go into it today, but some of the things we've been looking at is that there is evidence that HbA1c is also different between individuals of different ancestry. So European individuals generally have on average a lower HbA1c and the non-Europeans have a higher HbA1c. However, if you look at the underlying glucose, that does not correlate. So I think we need to be better about defining HbA1c thresholds for genetic architecture and potentially age, because there's also an age effect that we're exploring right now. So I think it's a generally good marker for diagnosis. I think it's really practical and it works generally quite well, but I think we can do better than just applying it blindly, one threshold, the same for everyone, ignore everything.
Thank you. It was a wonderful talk. I think my question kind of relates to what you've just said about variable thresholds for diagnosis, but this high effect variant you have in your African Americans on HbA1c that isn't mediated by glucose, that was mentioned in healthy individuals. Does it have the same magnitude of effect in African Americans with type 2 diabetes?
So that's a really good question, and we haven't done that. So one of the things I'm doing right now is actually I'm trying to get a grant to study this, and one of the things I'd like to do is actually, I don't think anybody's done this before, but it's actually recall individuals based on genotype and a different thing, and actually get a continuous glucose monitor on them, because then you also circumvent some of the issues that, you know, I say the HbA1c doesn't correlate with fasting glucose, but what's happening in the postprandial state, I don't know because I don't have that measure. And what's happening in the diabetes range, does the glucose just overcompensate completely? And so therefore it doesn't really affect, for example, if they've got a diagnosis, their treatment. Having said that, although historically people have used HbA1c to target diabetes treatment, increasingly, it's not there yet, so it's just starting in the type 1 community, but people are increasingly using continuous glucose monitors now to better target treatment rather than just measures of HbA1c. I think we're still a long way from doing that for the whole population of type 2 patients though, so I think it's a really good question that I think needs looking at.
Thank you.
Thanks. That was awesome. I was interested in the 80% of loci not showing significant effects of differential effects across ancestries, and then squaring that with the fact that polygenic risk scores built in one ancestry don't work anywhere near as well.
Yeah, I think that's because at the 80% that don't have effect allele heterogeneity, they're the ones that are genome-wide significant. And I think those that are genome-wide significant tend to be closer to potentially the actual causal variants that are probably the same across ancestries. I think some of the polygenic risk scores, as we incorporate more and more variants, they're going to be, some of them, not very good proxies for the actual thing. So, and I think because the LD is different across the population, you're probably exacerbating the differences between the populations.
have you tested it? Because if you build a score just with the ones you say don't differ, because the other possibility is you're underpowered to detect the differences that really are there, and those little differences add up.
Yeah, it's possible, especially because we do have differences in sample size. Yeah, it's true.
Hi, thank you for the great talk. I was wondering, when you were clustering genetic variants, how did you account, or how did you avoid capturing kind of ancestry?
So for the clustering, I believe at that point we only used European data, partly because we didn't have association results for all the other traits across all the other ancestries. So it's biased towards a classification based on European associations for other traits.
Sure. And within the European ancestry, was there any control for structure within those individuals?
Only in the original analyses, yes.
Thank you.
Any more questions? Thank you, Nash, for the wonderful talk.